A warm greetings everyone. I am Anjana AM of 2nd MSc Microbiology and I am here to discuss about the topic microbial degradation of industrial solvents. Coming into the topic, first we shall go into an introduction of industrial solvents. Industrial solvents are nothing but chemicals which are used for the dissolving or extraction of substances in manufacturing processes. So they have various uses in the industry. The uses include construction, manufacturing, pharmaceuticals and cleaning. In construction, they are used for the carriers as for paints, varnishes and adhesives. Manufacturing, it is used for dissolving or extracting materials. Pharmaceuticals, it is used to solubilize the active materials and remove impurities from the mixture of drug. In cleaning, they are used for the cleaning of automotive parts, tools and other machineries, etc. So as they have so many uses, these industrial solvents also tend to have many toxic effects in the environment. The toxic effects include air pollution, which harm water quality, climate change, and not renewable. So these solvents used in the industries, they emit toxic gases and cause air pollution in the atmosphere, and also it contaminate the groundwater. These all contribute to the climate change, and also these are not renewable sources, so it get depleted easily. So, coming into the mechanisms of microbial degradation, the industrial solvents are biodegraded mostly by the microorganisms. For that, the microorganism exhibit certain mechanisms. So, we can just look into some of the main mechanisms of microbial degradation of industrial solvents. So, these mechanisms are metabolism of solvents, enzymatic reactions, and bioconversion. In metabolism of solvents, these industrial solvents are used as a carbon source or energy source by the microorganisms. So it can be degraded either through aerobic or anaerobic pathway. So in aerobic pathway, the complex molecule is break down into simpler ones utilizing oxygen, and in anaerobic degradation, the complex molecules or complex solvent is degraded into simpler molecules either through fermentation or any other anaerobic processes in the absence of oxygen. Coming into enzymatic reaction, it is one of the main and common method of microbial degradation in microorganisms. So in that enzymatic reactions, they use specific enzymes for the degradation of these industrial solvents like hydrolysis, which would break down the esters and amide bonds, oxidizers, which would add oxygen into the molecules, which would help in their degradation. Reductases, they transform the harmful compounds to less toxic one by reduction of that particular compound. Then the next mechanism is through bioconversion. In bioconversion, uh, what they do is that the uh, molecules are transformed into intermediates, or they are precipitated or absorbed into the microbial cells. In transformation into intermediates, these solvents are converted into some intermediates of a metabolic pathway, which should help in their degradation. In the precipitation and adsorption, activation of precipitation and adsorption, the toxic hormones are immobilized through adsorption into microbial cells, or it is converted into some solid forms which would be not toxic in the environment. Microbial degradation of perchloroethylene. Now we are looking into the degradation of specific industrial solvents. So the first one is perchloroethylene. So as the name suggests, it is a chlorinated industrial solvent and is used in various industries for dry cleaning, degreasing, textile processing and adhesives. So here the biodegradation of perchloroethylene, it is done by a mechanism called co-metabolism. Co-metabolism is a type of degradation where two compounds are degraded simultaneously in a metabolic pathway. So the degradation of second compound will depend upon the degradation of first compound. So here the degradation of this perchloroethylene, which is a halogenated aliphatic hydrocarbon, it depends upon the degradation or metabolic pathway used for the degradation of aromatic compounds by the microorganism. So this is known as co-metabolism. So perchloroethylene is degraded in by microorganisms using this co-metabolism method. So the enzymes involved in this co-metabolism mechanisms are dioxygenases, monooxygenases, and hydroxylases. The organisms involved include Pseudomonas fluorescens, Pseudomonas twisteri, Bulgaria cepacian, and Pseudomonas pitida. So these are the microorganisms which are capable of degrading the perchloroethylene into non-toxic compounds. Now we can look into the mechanism of degradation of perchloroethylene. So in this co-metabolism, it uses a dioxygenase enzyme that is produced constitutively as the metabolic substrate is consumed. As the substrate is consumed, this dioxygenase enzyme is produced constitutively. So it destabilizes the carbon bond in the perchloroethylene to convert it into an organic acid which can be further degraded into carbon dioxide and water. So here we can see PCE epoxidation. So tetrachloroethylene, PCE is also known as tetrachloroethylene. So here we can see the carbon have double bonds. By the help of the dioxygenase enzyme, it would add the oxygen molecule between these carbons, and with that these double bond is destabilized, and this tetrachloroethylene is further finally converted into trichloroacetic acid, which is an organic acid. Then after that, the complete mineralization of this trichloroacetic acid is done by beta oxidation, which would produce carbon dioxide, water, and chloride as the final products. So this tetrachloroethylene or perchloroethylene can be converted into carbon dioxide, water, and chloride by this mechanism of co-metabolism. Microbial degradation of acetone. So the next industrial solvent is acetone. So it is used in the production of plastic, fibers, and pharmaceuticals. It is also a common solvent for cleaning and degreasing applications. So acetone, it is a biodegradable industrial solvent, but it is toxic to microorganisms because it has a membrane disrupting ability. So it is considered somewhat toxic to the microorganisms. So it can be degraded through both aerobic and anaerobic methods. In the aerobic method, it can be degraded using carboxylation pathway. In the carboxylation pathway, the acetone is carboxylated to form acetoacetate. This acetoacetate is further cleaved to two acetyl-CoA molecules, and these acetyl-CoA molecules can enter the DCA cycle for further degradation. Then coming into the next method in aerobic degradation, this direct oxidation. So some bacteria, they can directly oxidize acetone to form acetate and carbon dioxide. Directly they can skip the carboxylation step, and they can directly oxidize this acetone to form acetate and carbon dioxides. In the anaerobic degradation, the organisms can use carboxylation pathway and formylation pathway. So this carboxylation pathway is similar to that of the aerobic degradation, where the carboxylation of the acetone is done to form acetoacetate, and acetyl-CoA are produced from that. But here the difference is subsequent steps may differ depending on the specific microorganisms and environmental conditions. So it may enter the DCA cycle or undergo any other, the acetyl-CoA may undergo any other pathway depending upon the organisms and the environmental conditions. In formylation pathway, acetone is formylated to form 2-hydroxy isobutyryl CoA, which is then further metabolized to acetyl CoA. This acetyl CoA can be incorporated into DCA cycle or beta oxidation or any other metabolic pathways. So the microorganisms involved in this degradation of acetone are Pseudomonas pitida, Rhodococcus rhodoporus, Desulphococcus biactus, which these all belong to bacteria, and Aspergillus niger and Penicillium chrysogenum, which belong to fungi. The microbial degradation of toluene. So the toluene is used in paint thinners, adhesives, and coating. It is also used in the production of chemicals such as benzene and xylene. So the microorganisms employ a variety of metabolic pathways to degrade toluene. The most common pathway involves the aerobic degradation, where oxygen is utilized to oxidize toluene into less toxic intermediates. The microorganisms involved in this are Pseudomonas putida, Rhodococcus, Rhodochloris, Multidoria sepacian, Mycobacterium species. These belong to bacteria, and fungi such as Phanerochaete Chrysosporium, Pyotrus austriatus. The key steps involved in the aerobic toluene degradation are initial oxidation, ring cleavage, and further metabolism. So, in the initial oxidation, the organism can have dioxygenase or monooxygenase pathway. In the dioxygenase pathway, toluene is oxidized by oxygenases, enzymes that incorporate molecular oxygen into the aromatic ring of toluene. This forms a cis dihydrodiol intermediate. Alternatively, monooxygenases can oxidize toluene to form toluene epoxide. The next step is ring cleavage, in which the intermediate compounds such as this is dihydrodiol or the toluene epoxide formed in the initial oxidation step are further metabolized through ring cleavage. The ring cleavage can be metacleavage pathway or orthocleavage pathway. In metacleavage pathway, it is the most common pathway where the aromatic ring is cleaved between carbon atoms meta to the methyl group, leading to the formation of catechol derivatives. Here, the carbon atoms in the meta position of the methyl group is cleaved, leading to the formation of catechol derivatives. And in this orthocleavage pathway, the ring is cleaved between the carbon atoms in the ortho position of the methyl group, resulting in formation of different intermediates. Further metabolism, the cleavage products are then converted into central metabolic intermediates such as pyruvate and acetyl CoA, which can be further metabolized through the carboxylic acid cycle to generate energy and biomass. This is the steps involved in the aerobic toluene degradation. So first, the toluene is initially oxidized to form intermediates, which are then cleaved by metacleavage or orthocleavage pathway and further metabolized into pyruvate or acetyl CoA, which can enter the TCA cycle to generate energy and biomass. The next is the microbial degradation of xylene. So xylene, it is an industrial solvent commonly used in printing, rubber and leather industries. It is also used as a solvent for the production of paint and varnishes. So in the degradation of xylene, the key enzymes involved are monooxygenases and dioxygenases. So here the oxidation can be initiated by the oxidation of one or two methyl side chains by xylene monooxygenases. These enzymes, xylene monooxygenases, are specifically present in the organisms that are involved in degradation of xylene. So it would initiate the oxidation of one or two methyl side chains of the xylene. Then the complete dearomatization of the product produced intermediates, that is catabolic or non-catabolic compounds, is done by catechol dioxygenases. So these complete dearomatization is done, and it is the xylene is metabolized. So the organisms involved are Pseudomonas cyanantha, Pseudomonas mandeli. So next is the microbial degradation of chloroform. So chloroform, it is commonly used in laboratory settings and industrial application as a solvent for fat, oils and alkaloids. It's also used in the production of refrigerants. So it is a commonly identified aliphatic compound in the contaminated soil and groundwater. So it is a common pollutant of soil and groundwater. So the microorganisms involved in the degradation of chloroforms are dehalobacter, which use chloroform as a terminal electron acceptor during organohalide respiration. So this is the proposed pathway for anaerobic degradation of chloroform. So in this, what happens is that the chloroform is CHCl3. It is converted into CH2Cl2, which is which is a compound known as dichloromethane, which is less toxic when compared to chloroform. So the CHCl3 is converted into CH2Cl2 by dechlorination. So the reducing power needed to dechlorinate is produced from the fermentation of the DCM to carbon dioxide. So further, the DCM is converted into the produced DCM from the chloroform is converted into carbon dioxide or carbonate ions. And if there are any excess electrons after the reduction into DCM, they are used for the production or synthesis of hydrogen and or organic acids. Methane is another possibility that can be produced through this biodegradation of chloroform, but it doesn't happen because chloroform strongly inhibits the methanogenesis. Next, we can look into the factors affecting the microbial degradation of industrial solvents. So many number of factors, and some of the important or main factors that affect the microbial degradation are microbial community composition, nutrient availability, environmental conditions, toxicity of solvents, salinity, and biofilm formation. So microbial community compositions, the degradation of the industrial solvents by the microorganisms will depend upon the composition of the community in that surroundings. If the composition or the community has microorganisms that could specifically degrade the industrial solvent, it would enhance the solvent degradation, and the solvent would be completely degraded, fastly it would be degraded. Also the bioaugmentation potential. So whether the community has organisms that would degrade the industrial solvent, or they have that much ability to degrade the industrial solvent. So these would affect the degradation of the industrial solvents. Next is nutrient availability. So if the environment or the surrounding has sufficient macronutrients and micronutrients only, the degradation of the industrial solvents could be possible. So the macronutrients such as nitrogen, phosphorus, etc. are required for the growth and metabolism of microorganisms. So then only if the macronutrients are present in sufficient amount, then only these microorganisms can degrade the toxic compounds. Also, these micronutrients such as iron, zinc must be needed for some activation of enzymes, or it can act as some coenzymes in certain metabolic pathways, etc. So the nutrient availability of the surrounding also affect the microbial degradation of industrial solvents. Then coming into the environment, they are important that the surrounding or the environment should have the optimum temperature, pH, and oxygen availability. For the aerobes, it would need more oxygen. Anaerobes should need no oxygen. It can do degradation in the absence of oxygen only. So these conditions would affect the degradation of industrial solvents. Optimum temperature is required, optimum pH is required for the best degradation of the industrial solvents. Then coming into the toxicity of solvents, it involves how much concentration of the solvent is present in that particular area or in that particular surrounding, and also the structure of the solvent. That is, if it is easily accessible by the microorganism, or if it is the structure is easily degradable. That all would influence the degradation rate of the solvent. Then salinity, if in some organisms cannot degrade compounds or cannot do their metabolism in high saline environment. So in such conditions, halophiles should be needed for the degradation or like degradation of these industrial solvents, etc. Then is the biofilm formation. So it is another factor if the microorganism is able to form the biofilm or not. So if it is able to form the biofilm, it can protect the biofilm can protect them from any other environmental stresses and can also increase the degradation of the industrial solvents uh, when compared to the microorganism which does not have any biofilm formation. Now let's see about some of the benefits of microbial degradation. So microbial degradation is a sustainable solution. It has reduced environmental impact. It is a cost-effective method, and ecosystem restoration can be done using the microbial degradation. So the bioremediation offers a sustainable solution for the industrial solvent pollution. It reduces the environmental impact by minimizing hazardous waste, and also it is a cost-effective method compared to traditional methods. And bioremediation can restore the ecosystem, supporting biodiversity and ecological balance. So these are some of the benefits of using microorganisms for bioremediation. So these are my references, and thank you.